Today we will discuss about what are the effects that occurs when calreticulums are expressed on the cell surface under oxidative stress in the melanocyte cells. In the previous video we have discussed how these calreticulum are translocated from endoplasmic reticulum to the cell surface under oxidative stress in the so what we have understood is that the PERK signaling pathways helps in the translocation of the calreticulums to the cell surface from the endoplasmic reticulum under oxidative stress. So in this video we will discuss what are the effects that occurs when the calreticulum are expressed on the cell surface. So the hypothesis that they have from the patient samples is that the immune cells are found to be immune cells, or more specifically CD8+T cells, are found to be higher in the vitiligo patients compared to the healthy patients. So the hypothesis that the calreticulum that are expressed on the cell surface could be responsible for triggering these CD8+T cells or boosting these immune cells. So they check the skin samples for the CD8 plus cells. In healthy skin they see very minimum CD8 plus T cells across the melanin. However, for the vitiligo lesion they see more CD8 plus T cells. The signals are significantly higher compared to the healthy skins. And similarly, for these vitiligo PL skins, also they see the CD8 plus CD8 plus cells are shown, and they are higher than healthy skins. When they quantified this immunofluorescence staining, they can see that the results are statistically significant compared to the healthy skins. However, the results are not statistically significant between vitiligo skin and vitiligo PL skin. So this indicates that in the in the this vitiligo patients or patients that are affected with vitiligo has high immune response, and this immune response could be because of the calreticulum. That is what they have hypothesis. So in order to validate their hypothesis, what they have done is that they have used these PIG1 cell lines with this uh, control short interfering RNA against the calreticulum, and also this control short interfering RNA, so it does not target any specific gene. However, this calreticulum short interfering RNA will target the calreticulum gene. So in this case the they check the signals for the CFSE, which indicates that CD8 plus T cells formations, or which is the indications for CD8 plus T cells. So what they have find out is that when they use these PIG1 cells with this PBMC is the blood cells that are derived from the patients. So the blood cells will convert it into the immune cells against these PIG1 cell lines. So when they use these PIG1 cells with PBMC, they can see that signals for this CFSE. And when they use these PIG1 cell lines with oxidative stress and the control siRNA and also the PBMC, they see the similar signals, which indicates that the CD8 plus T cells are formed in this case. However, when they use this calreticulum siRNA, that will knock down, or that will prevent the translation of this calreticulum. In that case, they see the reduced signals indicating that the calreticulum might play a role towards boosting this immune system or CD8 plus T cells. And the quantifications of this flow cytometry analysis shows that the results are statistically significant compared to the isotype when they use this PIG1, in which the control siRNA is used, and along with that the oxidative stress is introduced. However, when they use this CRT siRNA and oxidative stress, they see the reduced signals, and these signals are statistically significant compared to the previous case. And after that, they also check the dose dependent effect on the CD8 plus T cells with different concentration of oxidated H2O2. that will lead to a different oxidative stress, and they see that increase in the CD8 plus T cells with increase in the concentrations to some extent, but they do not mention the results why there is a decrease in the CD8 plus T cells beyond a certain level of this oxidative stress. And after that they check whether this migration of the CD8 plus T cells will also occur when they use the CRT siRNA, and they can see that when they use the CRT siRNA along with the oxidative stress, the migrated CD8 plus T cells reduce significantly compared to the case where they use oxidative stress with this control siRNA or no siRNA. So this indicates that it's the calreticulum plays a important role towards activating or boosting the immune response. And after that they check the one more cytokines. They check the one cytokines, that is IFN-Y, which are usually found to be high in the patients that are affected with the vitiligo lesions, and they can see that these IFN gamma levels are statistically significant in the case of control siRNA with oxidative stress, and the signals are reduced when they used CRT siRNA with the oxidative stress, indicating that these results are in line with the patient's observations. And after that they also check this IFN gamma and CD8 plus cells, both positive CD8 plus cells and IFN gamma positive signals in the flow cytometry, and they can see that when they, in the case of this control siRNA, they can see the cells, some of the cells are both CD8 plus positive and IFN gamma positive. However, when they use the control siRNA, there are very less populations of the cells that are CD8 plus positive and IFN gamma positive, indicating that the calreticulum on the cell membrane helps in this IFN gamma cytokine formations, or helps in the maturation of the CD8 plus cells to have IFN gamma positive cells, or immune cells should have, or the immune cells have both CD8 plus positive and IFN gamma positive. That is being shown in this figure, middle figure of the panel F. And when they quantified these results, it is clear that control siRNA with oxidative stress has high CD8 plus cells, CD8 plus IFN gamma positive T cells compared to the other case, that is where they use CRT siRNA in oxidative stress. So this indicates, and the results for the CRT siRNA with oxidative stress is not significant compared to the isotype or the control case, indicating that it's the calreticulum on the cell surface plays a role towards immune boost in terms of generating more CD8 plus IFN gamma plus T cells. So from this figure they made a point that calreticulum on the cell surface could boost the immune response in terms of CD8 plus cells, CD8 plus IFN gamma plus T cells. And if you need to understand more about this experiments in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.